Welcome back. Now that we have discussed the structure and function of both DNA and RNA, we are going to move on to proteins, which is the whole point of this entire process. DNA stores the information, RNA carries the information in order to make proteins, which actually are what do the work in your cells. So, a little bit of review back from Unit 3 earlier when we did organic molecules. Proteins are made up of amino acids. The building blocks are amino acids. There are 20 amino acids. Okay, so think about these 20 amino acids as like letters in the alphabet. So our alphabet, there are 26 letters, and those letters can be put together in specific orders to form words. Well, these amino acids can be put together in certain orders to form proteins, or what we call polypeptides. A protein can be made up of hundreds of these amino acids, but they have to be in a particular order, and then they're folded into a specific three-dimensional shape. So I want you to specifically remember this back from when we talked about organic molecules. The shape is the most important thing about an organic molecule. That is what makes it do what it does. So if a protein doesn't have the correct shape, that means it's not going to be able to do its job properly. So the genetic code and how we get from DNA to RNA to proteins. So we already know how we get from DNA to RNA. That's what we talked about last lecture in transcription. Now we're talking about translation. So if you think about what this word actually means, translation is going from one language to another language, right? So you can translate from English to Spanish or Spanish to English or, vice versa, whatever. Well, translation, in this case, we're going from nucleic acid language, so that's DNA and RNA. So we have nucleic acid. Those are DNA and RNA. And we're translating into protein language, okay? And the way we do that is by reading the genetic code that's in the mRNA. The genetic codes is read by a series of 3 letters in the mRNA. So starting from the beginning, there's a specific start codon. We call these 3-letter codes, we call them codons. So every 3 letters in the mRNA codes for one amino acid. So some amino acids have more than one codon, but the start codon, so every single translation is going to start with AUG. That's the first codon that's going to be read in an mRNA molecule, and that is going to begin the process of translation. So what happens is that the codon is read in the mRNA, and then we talked about tRNA, which is technically what is reading the mRNA, okay? So if this is your codon, AUG, the correct tRNA molecule that matches up to this is going to bring the correct amino acid with it and start a protein chain. In this particular case, AUG is always going to code for the amino acid methionine, or MET for short, which means that every protein chain is also going to start with methionine. So this happens in the ribosomes. The ribosome is going to read the first codon. The tRNA is going to bring that amino acid in. And then the ribosome is going to go on to the next codon, and that amino acid will be brought in. And then it'll go on to the next codon, and that amino acid will be brought in. It's going to keep going like that until it reaches one of 3 stop codons. All that is, it's a signal to finish the process. There's no amino acid that comes with those codons. It's just a signal to stop the process of translation. This is our codon chart. This is how you read the mRNA, okay? So like I said, mRNA, we have 4 letters, 4 bases, A, U, C, and G. Remember, this is RNA, so there's no more T, it's a U, okay? So on this chart here is every single possible 3-letter combination of mRNA and the corresponding amino acid. So you always start reading here. So remember how I said there was a start codon? The start codon is AUG. So let's say we're going to find, well, what does AUG code for? Your first letter, A. So it's going to be somewhere in this row here, somewhere in this box, okay? Your second letter is at the top. Second letter is here. That's your U, okay? So now it's going to be reading down, over. Now you can use the third letter here on the right, that's a G, or you can just look and see, okay, AUG, oh, that's what I'm looking for, and it codes for methionine or the start codon, okay? So anytime you have 3 letters in mRNA, you can figure out what it actually codes for just by using this process. So it's first letter, second letter, and then the third letter if you want, or you can just figure out where it is in that box. So there are 64 possible combinations, and every single one of these is on this chart. You do not have to memorize this chart, okay? I'm going to give you one. It will be posted in Schoology, so anytime I ask you to perform translation, you'll have this out next to you so that you can look up the 3-letter codons together. Now, again, those stop codons, you've got here UAA, UAG, and UGA. Those particular codons do not code for an amino acid. That's just the signal to the ribosome to stop translation and that the protein is done. So here we go with tRNA and anticodons. This is a little confusing. So we have codons in mRNA. So remember, the mRNA, the messenger RNA, is what was made in the nucleus by the DNA. That's the process of transcription. Now the mRNA is outside the nucleus. It's at the ribosomes. And this is where the tRNA comes in. So those codons that you have, so like for example, that start codon, AUG, okay? What happens here is the tRNA has something called the anticodon, which is the complementary base pair of the codon, okay? So that's how the tRNA is going to recognize which amino acid to bring. So again, complementary base pairs, remember we're still talking about RNA. So A is going to pair with U is going to pair with A, and G is going to pair with C. So UAC is the anticodon that goes along with AUG. So this particular molecule, this tRNA, has a 3-letter base called the anticodon on the bottom. And it's kind of like this little, it has like a little hairpin shape almost to it. So it's like down at the bottom and it comes around like this and almost looks like a little T or a cross. So the anticodon is going to be down here because that is actually going to temporarily bond to the codon. And then up at the top here, there is an amino acid. So this tRNA molecules are just floating around in the cytoplasm of the cell, and what it's going to do is this anticodon is going to bond itself to the codon, bringing the proper amino acid with it. And then the ribosome will move on to the next codon, and the next tRNA molecule will come down and temporarily bond, and they'll be temporarily bonded long enough to put those 2 amino acids together to make it a polypeptide. So here's what a tRNA kind of looks like, okay? Again, base pairs are exactly the same. They haven't changed at all. The only difference is that there is no T at all because this is RNA bonding with RNA. So A pairs with U pairs with A, C pairs with G pairs with C. So if your mRNA molecule, so here's your mRNA, if your codon is GCC, your anticodon is going to be CGG. It's the complementary base pair of that codon. And then up here, connected to the other side of the tRNA molecule is your amino acid. So every single tRNA molecule that has CGG as its anticodon down here is going to be carrying the exact same amino acid. And then once it drops it off, it can go pick up another one and it can bring it back. And it can go pick up another one and it can bring it back. So the actual steps. we've got a ribosome, okay? I'm not really going to get into the specifics about the P site and the A site and the large subunit and the small subunit. This is your ribosome, okay? Simplified version. So the mRNA threads itself through the middle of the ribosome, okay? And like I said, it's going to start reading the mRNA when it finds an AUG. That is the start codon. So once it finds that start codon, it's going to bring, so the AUG is your start codon in the mRNA. The ribosome reads that. And then the first tRNA molecule with the anticodon carrying, and the tRNA with this anticodon, so AUG is your codon, UAC is going to be your anticodon. So the tRNA having the UAC anticodon is going to bring methionine. It's always going to be carrying the same amino acid. We abbreviate MET. And it's going to bring and bond to the AUG. So these are going to bond together, bringing the methionine with it. And then the ribosome moves to the next 3-letter code, okay? Once it moves to the next 3-letter code, it's going to read that codon. And the tRNA with the correct anticodon is going to bring in the correct amino acid. And then those 2 amino acids are going to be bonded together through peptide bonds, which is why proteins are called polypeptides. The ribosome is going to keep moving along like that, reading every 3 letters. So it's not going to skip any letters in between. Once it finds that start codon, it's going to keep reading 3 letters, 3 letters, 3 letters right in a row. There's not going to skip anything until it reaches a stop codon. And when it reaches a stop codon, that's going to be the ribosome's signal to stop translation and to release all of those amino acids that are now chained together. So this is kind of just a little diagram of what's going on. So we start up here, our start codon. And this green thing here is our tRNA molecule with the correct anticodon. So notice here, we have temporarily bonded AUG with UAC. The codon is in red and the anticodon is in green. That brings, once they're bonded together, you have the amino acid. In this case, it would be the amino acid MET. And then the ribosome is going to move down the mRNA. Okay, so now we have another tRNA molecule coming in. It brings an amino acid, and as it's moving around, you see it's making a chain of these different amino acids. Okay, so as this one comes in, this amino acid is going to be added to the already growing chain of amino acids, and it's going to keep going. It's going to get larger and larger and larger, putting these together using peptide bonds until eventually it reaches a stop codon, and what you end up with is a polypeptide. We are going to do some review of this in our next class, but your assignment for today is to complete the Ed puzzle. The class code is listed in the assignment. So you just go to the website, edpuzzle.com, you put in the class code, complete that assignment. It's a video. It's going to stop and ask you specific questions along the way. And then we will come back to protein synthesis and get a little bit more, do some practice problems in our next class.